sea, ASEICA for You es un programa que está coordinado conjuntamente con ASEICA Mujer y ASEICA Joven, Marisol coordinando ASEICA Mujer y ASEICA Joven, pues Marisol y yo, pero aquí tenemos que mencionar también a Cima Moreno, que ha estado activamente implicada en todo este programa, y también a Patricia González Santamaría y Ana Yannick, que han trabajado muy duro en lo que es el programa mentoring, que ahora os quiero presentar. Entonces, ¿qué es ASEICA for You? O sea, ASEICA for You es un programa transversal que está pensado, ideado, pues, para potenciar el desarrollo y el liderazgo de los socios de ASEICA, ¿vale? Desde las fases iniciales de su carrera profesional hasta los puestos de alta responsabilidad. Por eso es transversal, ¿no? Entonces está dirigido tanto a estudiantes predoctorales o incluso estudiantes de máster que sean socios de ASEICA, a postdoctorales y a investigadores principales, tanto los emergentes como los consolidados, ¿no? Entonces, este es la idea semilla de este programa ASEICA for You. Entonces, en este programa, de momento, nos hemos fijado dos objetivos básicos. El primero de ellos, que llevamos trabajando en él pues, desde que surgió ASEICA Joven, pues, allá por principios del 2018, ¿no? Esta idea en la cabeza que teníamos de establecer un sistema de emparejamiento en el que mentores y mentoras en el área de oncología, ¿no? De socios de ASEICA, asesoraran a investigadores que se encuentran en las distintas etapas de su trayectoria, ¿no? Este es el programa mentoring. ¿Vale? Y el segundo de los objetivos es la organización mensual de talleres y webinars muy interactivos para mejorar el desarrollo, ¿no? Para intentar dar un impulso, ¿no? Al desarrollo profesional de nuestros socios. Y en este objetivo se contextualiza el Meet the Expert, ¿no? Entonces, el Meet the Expert es el primero de estos talleres o webinars, pero estamos ya pensando en siguientes que creemos que van a ser muy útiles, ¿no? Para todos los socios de ASEICA. Entonces, os quería explicar en 2 slides el programa mentoring, que ya pues, está ya hechos bastantes emparejamientos. Entonces, empezamos en abril y septiembre, en el pico de la pandemia, enviamos una encuesta a todos los socios para saber el interés que había, ¿no? En este programa y si los socios, ¿no? que querían participar, ¿no? Tanto como mentores como mentees. Entonces, aquí os pongo un poco los números. O sea, al principio tuvimos mucha aceptación, mentees más de 90 y mentores 50. Entonces, después de este Monkey Survey, lo que hicimos fue preguntar a los mentorizados, a los mentees, qué mentores de estos 50 pues, les apetecía, porque ¿no? les gustaba más. Entonces, hicieron una selección. Entonces, en esta selección participaron de los 90, 54 mentorizados, aquí como veis, y luego de los mentores 51. Para que veáis un poco la distribución de los mentores, había investigadores postdoctorales, poquitos, creemos que se tienen que apuntar más porque pueden mentorizar perfectamente, a IPES Junior, ¿vale? Que eran los mayoritarios, y bueno, IPES Senior 21. Y luego mentorizados, pues había un poco, los mentees había un poco de todo. Entonces, con estos números, lo que hemos hecho desde octubre, que ha sido una tarea ardua, pero bastante satisfactoria, la verdad. Entonces, hemos hecho 50 parejas ya. Entonces, a partir de esto lo hicimos ya la semana pasada, acabamos con los 50 emparejamientos. Entonces, si alguno de la audiencia estáis ya en el programa mentoring, que sepáis que os vamos a contactar en breve, ¿no? Para presentaros a vuestra pareja en el mentoring. Entonces, os quería mostrar, por ejemplo, 3 ejemplos, porque esta es la dinámica que nosotros hemos seguido. Creemos que hemos formado parejas en los que un IP senior mentoriza a un IP junior, en este caso, pues Arcaich y Patricia. Luego, un IP junior va a mentorizar a un postdoc, ¿no? Verónica y Carlos. Y Amaya Ercilla, como postdoc, pues va a mentorizar a una prelo, que es Ana hoy también. Entonces, para que veáis que esta es la dinámica de emparejamientos que hemos seguido, ¿vale? Y seguiremos. Esto va para largo. Y ya para finalizar, os quiero recalcar otra vez, ¿no? El objetivo de este programa, que es potenciar la interacción entre socios para ayudar al desarrollo, ¿no? De todos los socios en cuanto al liderazgo y el desarrollo profesional, ¿no? Según cada uno esté en la etapa. Entonces, con esto os dejo. Vamos a escuchar a Salva, que nos va a contar cosas súper interesantes. Muchas gracias a todos. Ahora, ok.
Pues nada, muchísimas gracias, Verónica. Como veis, este es un programa al que hemos puesto mucha ilusión y yo creo que va a funcionar muy bien, ya veréis. Y nada, seguro que salen más pares y vamos a tener más media experts. So now I'm going to switch into English to introduce our speaker. And so this talk is going to be recorded, and it's going to be recorded because then it will be also online, and the other ASEICA members can benefit from what we're going to learn here. So, and then at the end, we will have, actually, we left quite some time for Q&A, so questions and feedback. This will not be recorded. And the questions you might ask, you can ask them in Spanish or in English, or how do you feel also? Okay, so now I'll get started with Salvador. And so, you know, we always say in these talks, so we are glad to have the speaker here, but really we are really, really glad to have Salvador. I think most of you know him. Is one of these kind of people that you could talk for hours, actually, because he has so many interesting things that he's actually developing. And for sure, and he will be involved in ASEICA for You, but also in ASEICA Responde, that, you know, it's another initiative that we have for outreach and the society. So you will see more of him during the year. But now just get into him. So Salvador has an interesting kind of curriculum, well, an interesting and long, so I'm going to short this like a 1 minute. Well, first he got his master's at the McGill University in Canada, interesting enough. And then he moved to the Fiona Watt Lab, which, you know, is a Cancer Research UK. And there he got his first papers in science and the level, working on stem cells, on RAC, on epidermal development. Then moved to the CRG first, and then to the IRB, where he's now, and he's an ICREA professor. So the CV is very long, and it has lots of words. I could go on and on, Salvador Ochoa, and many. But so just so you see who are we talking with. So he has, I'm going to read here. So 5 papers in Cell from his group, okay? 5 papers in Cell, 2 in Nature, 3 in Nature Cell Biology, 3 Cell Stem Cell, plus there are a lot of other collaborations. So, and opening really new areas of research, not only in stem cells and homeostasis and you know, cancer and so on, but also in circadian rhythms, which is quite interesting, and tumor initiating cells, many, many, many things. So also, and in addition, and why we have him as well here, he's an entrepreneur, and then last year he started with other people, Ona Therapeutics. The company has raised a lot of money. He's going to talk into that. And then not only that, so he's also a person that is very much in contact with patients and families of patients. He's going to talk about a beautiful initiative on for pediatric cancer. So with that, I'm not going to take more time. Here I'm giving you Salvador Aznar Benitah. So Salva, when you want to.
Okay, thank you very much. That was a very nice introduction, but wait, first let me share the screen. Okay, there you go. Yeah, thanks so much. Can you see it now?
Yes.
¿La veis? Sí.
Sí.
So thanks a lot. Muchas gracias, Marisol, and the rest of you for the invitation. It is a very interesting thing to give a talk like this. No, not the usual scientific talk, and where one can sort of give an idea of well, what moves you, to do some of the things that we do in our day-to-day job. I think you have, I had added this slide with sort of like what was my previous education and previous positions and current positions, but you have made that introduction very well, so I can skip that part. So I want to give you a little bit of an overview before I get into all these, the couple of initiatives that Marisol has mentioned, a little bit of an overview of basically what are the questions that we have in my lab. And I think if it has to be summarized in one sentence, it's basically how is tissue homeostasis maintained and why is it lost? That is really, I think, and because in fact, when I started my lab, I had quite a lot of criticism, I must say. Right before my first evaluation, I had quite a lot of criticism that people telling me that I was very dispersed. And I would always tell them, well, this is how I am. I don't think you can have your lab being different to the way you are dispersed, meaning that I was touching up on many different interests. And I guess it is true, but I never saw it that way. I always saw this as a unifying theme, which was tissue homeostasis and really why is it lost. So how are tissues maintaining their daily business, their daily physiology? And from that sort of daily physiology, you can kind of understand, well, why we're in circadian rhythms. How do stem cells, for instance, respond to the needs of the tissue? Sometimes they need to change their identity. Sometimes they need to, so for example, when in the skin, when there is a wound, hair follicle stem cells will change their identity in order to become epidermal cells, in order to contribute to the wound. So whereas epidermal stem cells, which are right next to the wound, they don't need to change their identity. So there's a lot of epigenetic mechanisms there at work. And I think that explains why we work so much on epigenetic mechanisms. And then what I mean by why is it lost, really we look at two main aspects of it. Aging. I'm really interested in what happens when we progressively lose the ability to maintain homeostasis, and transformation in cancer. So just for you, in case you are interested in this and you want to ask me about it, I will not get into the details, but we are putting a lot of effort in trying to understand this daily business. So through circadian rhythms, through these internal clocks. But this has become really fascinating for us in the last years because we've realized that circadian clocks are not only providing our time frame to each cell in our bodies, but it's also allowing tissues to synchronize their function. So it's a means of communication between different tissues so that they're all performing specific physiological functions for the organism in a synchronized manner. And that is being really, the first two papers are these last two in 2019, when we're starting to really put out this concept of communication and how this communication is lost during aging. Because in fact, we think it's one of the main or a very prominent problem in the process of aging. And well, basically, yeah, this is really just what I said. So what we know is that every tissue has an internal clock, and that clock maintains some of the basic functions that the tissue has to provide for the organism. Without that, basically the whole system would succumb. But there is and that's approximately, we know now that's like 12 to 14% of the functions of each tissue is purely autonomous. The tissue does not need anyone to tell them that they need to do this function. But the other 85% requires communication, systemic communication. And we've developed a specific mouse model that for the first time allows us to look at this communication between 2, 3, 4, 5 tissues of choice. So yeah, been quite exciting. Now, in terms of epigenetic mechanisms, we've done a lot of work as well, but I think if I would summarize, really, I think what we most have learned from this work, our work, and also from other people, that, well, contrary to what was the dogma for embryonic stem cells, in terms of epigenetic mechanisms, we now know that chromatin factors and epigenetic mechanisms are mostly dispensable for the adult stem cell identity or homeostasis. So you eliminate most of these factors and the stem cells seem to be all right, but unless you stress the stem cell. And of course, our stem cells are being under continuous stress every day. So it allows stem cells to remain flexible towards stress. We know that many epigenetic factors are, in fact, among the most mutated genes in cancer. So they really affect not only the onset, but also the progression of cancer. So we're trying to understand this quite a lot. And then lately we got into this paper here in Nature Cell Biology in 2018. We got into this first thing of looking at how these epigenetic modifications even affect the mutational burden and distribution of mutations in specific tumors. So it's a very interesting concept, how the mutation of one epigenetic factor will actually affect all the other, most of the mutations that the tumor will accumulate with time. So also, if you're interested in that, we can discuss this afterwards. And now in terms of cancer, I will just highlight what we are doing in the last years, which is really concentrating on metastasis. We had done previous work on cancer, also linking into circadian rhythms. We stopped all that once we published this paper, in which we had the luck, I think, because we really weren't looking for this. We were just looking at whether there was any cell cycle heterogeneity within the cancer stem cells of different tumors. And it was more, in fact, the hypothesis was nothing very conceptually very new. It was just thinking, well, if we can find in these different types of cancer quiescent, long-term quiescent tumor initiating cells or cancer stem cells, maybe these are the responsible ones for the resistance to chemotherapy, for insensitive to chemotherapy. Well, it took us quite a while, but we actually did find a way of seeing these long-term slow cycling cells in the primary tumor. But when we purified them, assayed them, and studied them, etc., they very strongly suggested that they were metastatic initiating cells. And in fact, that was, in the end, when we did the functional assays, that was the case. Now, one of the great things when you find a specific population is that for the first time you can ask, well, what makes you so different? What makes you so special compared to the non-metastatic counterparts? Because we know that within the primary tumors, this is a minor population, like approximately 3 to 5%. And one of the things that we saw is that they have a very prominent fatty acid metabolism. So much so that you can identify them now by the cell surface expression, very high cell surface expression of CD36, which is one of the main long-chain fatty acid transporters, also for oxidized LDL, HDL, LDL, etc. And this, of course, is very interesting from a research perspective, but it's also very worrying from the perspective of the way we eat nowadays, because that means that these cells are very sensitive to not only the levels, but mostly the type of fats in our diet. And I can tell you now that mostly it's about palmitic acid. We've assayed many of them. There's a paper that we have right now under review. So unfortunately, I cannot tell you much about it. Hopefully I will be able to tell you much about it soon, but it means that they're very sensitive to diet. So looking at how much palmitic acid we consume in supermarket food, of course, it brings on a very worrying sort of link between diet and metastasis. Now, the good side is that they are kind of hooked on fat. So if you stop the ability of these cells to eat ingesting fat, if you knock out CD36, knock down CD36, or inhibit its fatty acid domain, or I will show you now, if you systemically inhibit with neutralizing antibodies, this is a very good therapeutic target. One thing that is very important is that we started our work with oral cancer, oral SCC, luminal breast cancer, luminal B, and melanoma. But we've now seen, we and others have seen, as I'll show you I don't have that slide anymore. I think I skipped a slide that was important. Well, basically I can tell you that stuff. So it operates in many human tumors. It's been very exciting in the last 3 years. I can tell you it's triple negative breast cancer. Melanoma has been conferred by other 2 groups, glioblastoma, sort of the intra-brain invasion, pancreatic, prostate, gastric, kidney, bladder, and then also in leukemia, chemoresistance in leukemia. And what has been very interesting, I can tell you also very interesting for the investors of Ona, is that 3 recent papers have shown that there's a very strong synergy between anti-CD36 therapy and immunotherapy. And this is in fact a role of CD36, not so much in the tumor cells, but in the immune compartment of the tumors. So of course that opens the possibility that whether the tumor cells express or not CD36, it doesn't really matter anymore, because pretty much all immune infiltrate in every single type of tumors actually have a metabolic reprogramming, part of it relying on CD36, and that gives a very interesting, very strong synergy effect with anti-PD-L1 and CPA as well. So I just want to give you a very just clear one of the examples. We've tested this in many tumor mouse models now, but this is, for example, you treat with a neutralizing antibody. And what we do is we do a therapeutic setting. What I mean by that is we know that inhibition of CD36 it completely eliminates the ability of the tumor cells to initiate metastasis. So that's a very strong effect. But of course, this is very difficult to control in the clinic, unless you would have a preventive treatment. So of course, most of the times the patients already come with a metastasis. So here what we do is we inoculate patient-derived tumor cells, whether it's oral, melanoma, breast, all the ovarian, also serious ovarian cancer, very strong effects. We wait for the primary tumors and the metastasis to grow, and then it's when we start treating with the antibody. And what you see in most of the cases, it varies from tumor to tumor, is that, for example, you see you start to have already a metastasis here in this oral cancer. You start treating, and this metastasis has completely disappeared. Whereas, for example, the primary tumor doesn't really care that much about the therapy of CD36. Now, this complete remission will occur in 20-25% of the cases. And then in the other 80-90% or 80% of the cases that don't show complete remission, the growth of the metastasis is blunted by approximately a 90%. So it really, if you think about it, is a dramatic anti-metastatic effect just by inhibiting this factor. So based on all this, in May from last year, we founded a company. Of course, I had started doing this, I would say, in early 2018. So it took me like a year and a half to really get to the point of having the money to fund it. But and the point of Ona is basically to develop therapies based on this. You can go to our webpage and see basically what is and this is actually the slide that I wanted to tell you about. Why this one is important, because this actually shows you. Sorry? Oh, I don't know. I heard someone. So this actually tells you how this paper, I would recommend all of you, if you're interested in it, they did a very thorough computational analysis in which they looked for commonalities linked to metastasis, irrespective of the tumor type. And what they came down to is that, well, what the title says, genetic alterations in fatty acid transport and metabolic genes are associated with metastatic progression in general in human tumors. And they managed, actually, we tried to do that, and we didn't get, we got down to an 18 gene signature. They managed actually to bring it down to 5. So with this 5 gene signature, they can basically predict poor prognosis and metastasis in most tumor types. And as you can see, one of the genes is CD36. So we were, of course, very happy as that confirmation. In fact, from this 5 signature, caveolin CD36, CPT1C, and CYP2E1 was already part of the signature that we had defined before. And this is what I was telling you about. These are all the tumors in which not only correlations have been established, but functional proof that if you inhibit CD36, you have a very strong anti-metastatic effect. We were part also of the ovarian serous cancer. All the others have been published by others. Well, we also showed melanoma and luminal A and B, but others have confirmed that. And this is where for example, the melanoma therapy resistance, pancreatic therapy resistance, and also this is what I was telling you about the immunotherapy synergy, is regarding T regulatory cells, CD8 cytotoxic cells, or also the transition of M1, M2 macrophages, if this nomenclature is allowed anymore, and natural killer cells. So now at Ona, as I said, we started it in May 2019. I found sort of like my other part of the orange, which is Valerie Van Horen. We had, I initially contacted her as an advisor because, of course, you know, the investors were asking me things that I had no idea what they meant, and I didn't understand anything. And so she has a lot of business background. She worked in a very important company in Belgium. She's from Belgium, who got sold. It's Ablinks, who got sold to Merck by enormous amount of money. She gained that a lot of experience. So we coupled together. We saw that we get along very well professionally and personally, and so we co-founded Ona. We have lots of people involved right now. We have, of course, very importantly, our clinical advisor, which is Josep Tabernero. It's amazing to work with him. Every time we have meetings, which tend to be at 5:30, 6 in the morning, by the way, because that's when he has the time to meet. His insight is really, really amazing, you know, in terms of how to shape initially the clinical trial, which type of tumors, how to do it, etc. And this is basically part of the team right now. We have a COO, we have a research director, a CFO, a chief financial officer. Of course, we put out a lot of new patents. So we have an IP lawyer, which is very important. And we have our corporate lawyer, which is Francesca Ranera, who's absolutely essential for the day-to-day functioning of the company. So it's a team that is, as you can see, very international. It is very experienced, multidisciplinary, that is absolutely essential, and of course, complementary. If you just look at where all these people have worked, it is really amazing, their background and what they bring in order to get, the idea is we want to get the best possible therapy to the clinic. And then the clinic will tell us, that's what the clinical trials are for. But before that, we want to do the best job possible in order to ensure that whatever we test as a clinical trial is the best possible therapy that we want to develop. So these are all the people that are involved. This is the actual team that works in the lab. We have, I have my lab at the IRB, which is in the Barcelona Scientific Park, and Ona is in a different quarter. So, and this is a wonderful people that work in Ona. This is Valerie here, and these are all different postdocs, technicians, you know, that they're doing some work in the animal model, some works in the in vitro testing. This is Bea, who's the chief, the science director, the COO. And then this is our latest recruitment, Salva. It seems that if you work at Ona and you're a man, you have to be named Salva. So, and it's great. He's helping us a lot with, you know, the protein production, etc. Now, just as a summary, basically, as I said, 2019, the way we started it was with a seed round of investment of 1.5 million. This was done by Asabi's partners. They're amazing. You know, they really put all the trust in us. Now, we thought of, instead of, which normally is what I think in these spin-off or startup companies, what happens is that you try to get financing through the different trenches, you know, sort of different milestones that you may have until you reach the clinical trial. We decided that we didn't want to do that because that takes a lot of time. You're constantly speaking to investors, and of course, I have a lab, you know, and that was taking quite a lot of time. I wanted to devote, Valerie and I, most of our time to the company, to the development of the product. So we set up a very sort of ambitious plan of saying, okay, within 1 year, we will get in a Series A investment, we'll get all the money necessary to reach the clinical trial, phase 1 to B, all right? And this, fortunately, even it was with the pandemic, etc., we managed to do this. It was intense, but really, really stimulating work. So now we have an international syndicate, which gave us 30 million. We closed this in July in 2020. This is Alta, Asabi's, and Isios, which are from Barcelona, and BPI, France, and Fund Plus from Belgium, which are really big international investment, all of them. And so if everything goes all right, which I touch wood, everything seems to be going quite all right, by 2023, we will start our first clinical trial. We already have our proprietary antibodies, etc. So, and the trial will actually be a basket trial. So because CD36 therapy works in so many different tumor types, we will not concentrate on one single tumor type, a basket trial of initially 6 tumor types, which we have almost already decided which tumors they will be. And then to finish, I wanted to tell you about this other project that I'm very excited about. And this, I just wanted to give it to you as an example of how important the collaboration, or at least I feel, how important the collaboration between basic research, clinicians, the patients and public donors can be and should be for research. Now, how does this all start? Well, there's this initiative at the IRB, which is called Crazy About Biomedicine. Now, Crazy About Biomedicine started, I think, 4 years ago. My lab has been involved in it every year. I really like it, and I like a lot to interact with these kids. These kids are amazing. This is 16, 17-year-old kids that come from, normally they come from the area of Barcelona, but they can come from any part of Spain, or any part of the world for that matter. So what they do is they spend their entire summer, Saturdays and Sundays, in the lab with people, with mentors. My lab has had every year 1 or 2 mentors, and which of course is a very big commitment by these mentors. It means that they're going to be every weekend coming to the lab to deal, you know, to mentor these kids. But the point is that they do real research. This is not a little booklet that tells you, you know, how many microliters you have. No, I mean, they're sitting down, they're looking at the project, they're understanding what the project is, they're doing research, and if the experiment fails, they see that this is really, I mean, how most of the times it is, no? So in this first year, we had, in the first year, a group of kids that I had in my lab. There was this kid here in the middle, Felix Junquera, who was he created a very big impact in all of us, and especially to me. He was very, I would define him as a light in a sense, you know. He was so thrilled and so energetic and so excited about this, and he was so inquisitive. No, the type of person you would think, wow, you know, this person is going to be an amazing scientist or an amazing whatever he decides to do in his life. He was very cheerful, and as you can see already from the picture, Felix was not doing well. He had a limp, you know, when he entered the lab. He couldn't walk well. That degenerated in a month or so, being in a wheelchair. And unfortunately, by the end of the Crazy for Biomedicine, he passed away. He had a very aggressive pediatric osteosarcoma that had spread to the lungs, and he couldn't go through it. So the parents of Felix got in contact with me, and for a year, we actually became very good friends. We still are very good friends. We meet with our families and my kids and the other daughter that they have. We go for lunch and dinner. We've created a very special bond. They are part of the group of families of San Juan de Deu they dedicate most of their time to find money in order to put money into research, because as you will see now, the figures are so poor, no? When it comes to pediatric tumors. So they initially told me that they want to give me the money, part of the money that they had raised for my lab. I said no, because I was too sort of like emotionally attached to them. And I told them that I would commit to find what could be the best lab to invest the money. And I'm not saying that my lab is the best at all. But what I am saying is that I found it very sad I spent a whole summer looking at the literature. You know, I would wake up early before my family did. And I found that there was basically nothing. There's just recurrent testing of cell lines that have been there for years with conventional chemotherapy, and there's nothing new, very little new real good research, you know, being performed as is being done with adult tumors. And so there I told them, well, let's link with the director of oncology of San Juan de Deu, Jaume Mora. He's the first person to actually establish a real pediatric cancer unit, which he got trained at Memorial Sloan Kettering. Is the first real pediatric cancer unit of research devoted in Spain. And we joined together, and basically the idea is, okay, Jaume, tell me what is the problem with these kids? And he immediately said, the problem with these kids is not the bone cancer. The problem is when it reaches the lung, we have very little to do there. So the idea is to study what are the causes that underlie pediatric osteosarcoma lung metastasis. As you can see, the outcome of these patients is, once they develop lung metastasis, is dismal. But the problem here, plotted this 35 years ago, it would have been exactly the same. Not a single new therapy, nothing, no research, no, just basically what has been happening in adult patients has just been tried in the pediatric, but nothing specific for pediatric osteosarcoma. And so the first challenge that we had was, can we develop from the patient-derived tumors as soon as it's obtained from the surgery or obtained from the surgery and frozen? Can we develop an orthotopic mouse model that would recapitulate what unfortunately is happening in the patient? And we're very happy that we did manage to get this. So we have implanted the tumor in the bone in the leg, the bone of the leg, where this tumor appeared also in the patient. And you can see already that some mice are developing lung metastasis in the same mouse, all right? So this now allows us for the first time to really compare primary versus lung, do all the panoply of analysis that we have in my lab, lots of single cell RNA sequencing with positional information, site of all the different therapeutic type of analysis. So the working plan is, well, identifying metastatic initiating cells. Are they the same as we have seen in the tumors of adult patients? Or maybe they're different actually in pediatric. Pediatric cancers and adult cancers are very different. You know, one is a developmental type of tumor. The other one is just accumulating lots of mutations with time. And in fact, here it is one of those cases where, of course, it is very important to publish in very high impact journals because money comes in from that, and you can it allows you to continue to do your research. But here, my main goal and the main goal of everyone involved in this project is to develop possible therapies. And we might fail, but at least we will try. At least it will be a very serious attempt of trying this. So, of course, the idea would be to patent and either development of this thing through a spin-off. Either might be Ona, might not be Ona, we don't know there yet. But the first phase, we're doing the study with 10 patients. That's already a lot of money if you think about all the single cell RNA sequencing, all the functional assays that we're doing. And the second phase is that we're also attempting at doing the analysis, not only from the orthotopic models, but directly from the surgical specimen. It's a bit more challenging, but we're confident that we will get this. So the idea would be to establish this as a approach, as a preclinical clinical pipeline, that as soon as a patient comes to the hospital, we could analyze the tumor in parallel, and then hopefully we could determine what would be the best type of treatment that patient should get. The annual cost actually of all this is not low. It's 200,000 euros a year. And this actually goes on to show this is being provided to date uniquely from the parents. So if any of us here in this audience would get a 200,000 euro grant per year, we would be very happy. But think of how many grants are out there for 200,000 euros to study a pediatric tumor that you basically cannot even show you have experience in. But you know that you can do it because you're joining with the clinicians who do have a lot of experience, and you bring your own, and that there's a synergy. So I think it's a very good example. Also, it's very stimulating. I tell you one thing, when we currently meet with the parents and the patients, and it's, of course, it's a very different way. It puts in perspective everything that you really don't care about impact factors anymore when you're dealing with them. So I think that is, I think that is the last slide. So I don't know, the last slide is this one. I also had a funny one about the clock of my hair. I'll show you now. So this is my wonderful lab. I have just a mixture of PhD students. So all of them are really amazing. A couple of research associates, which are very sort of like stable long-term postdocs, especially Gloria. She's the one who started the whole metastatic thing, and she's the one who's been the longest having to suffer me for the longest. And then there's a postdoc, and of course, very importantly, the senior technician and the bioinformatician, and the type of work that we do in my lab is really essential. And then just to finish, if you want to see why is another interest that I have in studying aging and circadian rhythms, well, this is a very relevant question that I want to know about, is what the hell happened to the clock in my hair since I started as a PI and only 7 years later. I mean, this is actually what it does to you. So hopefully we can find something to stop that. Anyway, I leave it there. Sorry.
Thank you, Salva. I think many men would, wouldn't mind to have not even your hair, but your whole kind of, body, I would say. Anyway,
yeah.
Thank you so much. I think this has been very inspirational talk, and we have seen how important it is to, I guess, to take risks, right? And also to join forces with other people. Pues nada, Salva, nos has dejado impresionados. Bueno, esto por supuesto ya lo esperábamos, pero todavía más. Entonces empiezas hablando de dispersión, pero luego lo que hemos visto es que realmente es la entropía, y aunque pasa por unas fases de ansiedad, pero esta ansiedad es capaz de canalizarla de alguna manera.
Eso es.
Bueno, viendo poco, pero arriesgándose y rodeándose de un montón de gente interesante, delegando, que ha dicho delegar, que me parece muy interesante. Pues nada, delegando y uniendo esfuerzos, asociándose a los pacientes, y yo creo que aprovechando las oportunidades, diría yo además, pues hemos conseguido este pack que bueno, que es único. Habéis visto en 5 años pasar de propiedad intelectual a un ensayo clínico. Eso es súper excepcional, así que te vaya muy bien. Ojalá. Y nada, te tenemos apuntados, por supuesto, estás apuntado en el Meet the Expert, en ASEICA Responde y
mentoring también, si quieres, ¿eh?